Today I'm going to be giving a presentation on the SYNJ1 gene, as well as a structural and functional analysis for how a certain missense mutation of arginine to histidine at the 457th amino acid position could be directly linked to certain neurological diseases, such as Parkinson's disease. Now, the SYNJ1 gene, which is short for the synaptogenin-1 gene, is a phosphoinositide phosphatase enzyme that is directly responsible for the excision of phosphate groups off of phosphatidylinositol lipids. Now, it does this through its two domains, the SAC1 domain and the 5-phosphatase domain. And through the process of that excision of phosphate groups, it actually drives certain cellular processes such as actin remodeling, moderating the curvature of the cell itself, as well as autophagic trafficking in neurons. Now that I've kind of given a backstory on what it is that this gene does, we can kind of look at what it can cause. So, Parkinson's disease, it is a progressive neurological disorder that causes dopamine producing neurons in the human body to begin to degrade. Now, this certain gene could cause Parkinson's disease through that arginine to histidine mutation. Arginine, which is a very strongly charged, very permanently charged amino acid, gets swapped for histidine, which is not only not as strong of a charge as arginine, but histidine also has a very pH dependent charge. So you change the pH of the environment, you change the charge that amino acid exhibits, which could be very detrimental when you're talking about something as complex and complicated as a biological system. Now, if you direct your attention towards figure 6, you can see that I first found the SYNJ1 gene by using simple ClinVar. I used their database to construct a variables of unknown significance chart, and then I selected, it's the one with an arrow and the blue dot in the middle of the black dot. That is the variant I selected, which is very close in proximity to a known disease-causing variant, and that's why I chose that variant. I then used that variant, I put that variant into Polyphen2, which gave me a probably damaging score and also the highest score you can possibly get for something that's probably damaging. So I then use the FASTA sequence to construct 3D models in USARA of my protein, which, if you look at figure 1, the green model is the wild type variant of my SYNJ1 gene. And if you look at figure 2, that's the mutant version. Now, they look very similar. They are very similar. Actually, if you look at figure 3, you can see that USARA gave me a 99.94% similarity score between the two models, which means the backbone and overall structure of the molecule is very conserved between wild type and mutant. However, you look at figure 4, it's a zoomed in image of figure 3, and it highlights the amino acid changes, and you can see that there is a complete change in the structure of the amino acids, which then can lead to malfunction of the protein. The protein stops working, or it works at a very inefficient rate, which is what leads to Parkinson's disease because it can't excise those phosphate groups as well as it should be able to. Now, to kind of wrap it all up, I would like to say that we need to develop further research into this gene variant, as well as we need to do further research into the SYNJ1 gene as a whole, because not only could further research into this gene potentially lead to cures for Parkinson's disease, or maybe even ways to prevent it altogether, it could also pave the road in understanding synaptic lipid regulation for neurodegeneration, which could not only cure Parkinson's disease, but also cure a vast, very broad scale of other neurological disorders. Now, before I close this out, I would like to give some acknowledgments. This research was conducted as part of the DNA Code project at Hudson Alpha Institute for Biotechnology. I would like to give a special thanks to Simple ClinVar, UniProt, Polyphen2, and USARA for allowing me to use their softwares and websites to develop this research project, as well as to obtain all this knowledge that comes with it. And I would like to give a special thanks to Sylvia, my TA, for giving me assistance and guidance throughout the course of this project.